I'm really pleased to be able to introduce Colonel Dan Wattendorf. Colonel Wattendorf is an active duty Air Force physician who is a geneticist as well as a family practice physician, both. And he's going to talk to us about his approach to revolutionizing the way we deal with infectious diseases. Thank you.
Thank you, Jeff. Good morning. When a fire occurs anywhere in the world, we cannot predict it today. It can occur by an act of malfeasance by an individual, and it can occur by a natural event like a lightning strike. And when that fire occurs, the reaction is immediate, and it's immediate because the fire spreads. And here we see an image of people clear-cutting across the forest to create a fire break to stop the spread of fire. The action needs to occur in the time that the fire is occurring to prevent that spread, and that action must be immediate. But our tools in infectious diseases are not like that. We don't have the ability to create fire breaks like we can in real time during a fire. So what we are trying to do inspirationally with these programs is to understand what are the tools in this era that we can create that act faster than our traditional methods of controlling an infectious disease process. And the tools that I bring to bear are a little bit of knowledge of genetics from the community that is at large here and others who have trained me. And so I have looked at the fields of genetics and gene therapy to understand how quickly can we act and what tools can we employ. So we saw a case example of this that we all lived through this year, and it was Ebola. Now, we knew about Ebola. Ebola is something that in the public domain, we need to be able to respond to Ebola much quicker than we did. But what we saw before our eyes showed that we did not have the tools today to act with a fire break that fast. So, Ebola, over a year ago, started in Africa, in a village on the west coast of Africa, and then spread immediately in country. And because of air travel and because of contact, was able to cross into Nigeria and then later into Europe and the United States. And because of epidemiologic controls, we obviously were able to limit quite a bit of those cases, and we are lucky in this case right now to see Ebola diminishing. But what I just want you to point to, or everyone to look at with this example, is that there was a long lag time before we saw most of the cases. And that lag time was several months, 4 to 7 months perhaps. But our responses, even now, are not in place in terms of our traditional methods of control. New drug or new vaccine. There's still no completed phase 2 vaccine, and there's no vaccine given to these communities that are at risk yet. And we are this many months after the outbreak. Is this the best we can do? So that was an inspiration that really occurred for months, for 4 years at DARPA of how do we deal with this threat. Can we go to the next slide? When we traditionally look at disease control, though, what we have done for eons is isolate people. When people exhibit physical symptoms of a specific disease, we put them behind a wall, and that wall is essentially the fire break preventing disease transmission. That was what was used today. Our ultimate solution that we prefer to have is a vaccine. But there's only about 24 vaccines present. The Ebola vaccine that people are seeing in the news, which is a viral vector-based vaccine, had been worked on for 2 decades. We were very lucky that this genetic strain of Ebola was consistent with the type of Ebola that we had been working on for decades. And we were very lucky to have done the antigen design and done some of the immune phenotyping to be able to get us to a place where we could leverage some of that to be able to provide phase 2 clinical trials now after the outbreak of disease has been transmitted. So it's too late. So 4 years ago, I was thinking, well, how can we do this faster? And DNA vaccines had been tried, and RNA seemed very compelling. There was a lot of research in RNA at the time, and I funded 2 companies, one Novartis, one Sanofi, a lot of academics, and a lot of biotech companies to try to understand could we make vaccines out of RNA. RNA, we would abort that process and be able to produce vaccines so much faster than we can with egg-based systems or tobacco-based systems. And some of those results have shown that we can produce an RNA vaccine at scale within a week with some of the people that are in this room. But we haven't designed the antigen. We have to go through an elaborate process of understanding what the structure you need to show to the immune system at what time with the right stimulation. And even when we give that vaccine, not everybody is protected, and it takes time to develop it. We all know about boosters. So are fast vaccines a solution? I'm not sure. I'm not sure it would work fast enough for this type of epidemic. There's another solution out there that predates vaccines, and that's serum transfer. In the age of diphtheria, the Iditarod, they took serum from horses that had been challenged with diphtheria. The horse generated immunity. They took the horse sera and then they brought it to Nome, Alaska to be able to block an outbreak of diphtheria. And it worked, but some people get sick from the horse serum. It's much better if you use humans, but even with humans, there's a risk of immune reactions, and you still have a fundamental problem of scale. It's 1-to-1. So we all saw Dr. Brantley do this. Now, you could argue whether how well it worked or didn't work, but what Dr. Brantley did when he recovered in the United States at Emory was he provided a blood sample directly to other patients who had been exposed and brought to the United States. And there was some factor in his blood that was going to be protective, and we know this factor to be an antibody. But that antibody is in a sea of other chemicals and antibodies, and it cannot scale. What we would love to be able to do is be able to scale the precise factor, and only that factor, that allowed Dr. Brantley to recover to the population at large, and we need to be able to do that very, very quickly. So I have 3 problems that I must solve to be able to accomplish this. I have to be able to isolate an antibody quickly. I need to be able to know which one is the correct one that is protective, and I need to be able to produce it and somehow give it to everyone in the population. So we would like a process in the future where we identify a disease in the world. There's unbelievable surveillance today because of genomic sequencing. There's unbelievable surveillance because of global communications, and identify those initial cases and employ our system. And in our system, what we would like to do is employ it in 3 to 6 months, and perhaps by the end of our program, prove that in people, not in papers, that we could accomplish this in 90 to 120 days, thus averting most outbreaks when the disease transmission is affecting most people. So conceptually, that's what we want to do. But how do you do this? How do you find these antibodies? We have colds all the time, and antibodies come up and they go down. You sometimes have influenza. Antibodies, again, come up and they go down. I need to find those antibodies when they're at a level of abundance, and I need to make sure the antibody that I find is the correct one. This is not, these are pretty slides. This is not an easy solution, even with today's best technology. So our process will be to isolate those individuals, collect their antibodies, and if we followed traditional medicinal biologics, meaning make an antibody, we would then have to deliver that antibody over and over again where there's a peak and trough drug concentration. There's only one antibody commercialized for an infectious disease as prevention of all of our biologics, and there's only about 30 of those. And that's Synagis, developed by MedImmune. It took them a decade to develop it. And they deliver that originally IV, and now can be given IM. But it is something that requires cold chain. You have to refrigerate it. For most of the antibodies that we would be thinking about, you would need to be giving IV doses. That's not scalable. And you'd have to give repeated doses. That is not a solution that works. So once we discover this antibody, we've got to deliver it in a different way. The punchline's already been delivered. We know that that would be genetic. But there's an aha here. I do not believe that we can provide genes to healthy people that are integrating with our bodies and permanently producing antibodies. some of the papers that you may have seen, particularly some of the people in this audience. We do not want permanent gene therapy producing an antibody against Ebola or any other given infectious disease in perpetuity for the rest of our life. That would not be a possible safe solution. We want to be able to have an ephemeral, a transient system. So we need to use a genetic system, but it's got to be transient. And of course, we go back to our RNA system because RNA is an ephemeral construct, or certain types of DNA delivery systems. And those are the ones that we've looked at. So after we've isolated the antibody, rather than putting them into traditional biologics that are made in silos and takes a year to produce an antibody, and we work with the pharmaceutical companies that make Synergists, such as MedImmune. It's a year-long process to make a mammalian cell. We are going to bypass that and immediately take the genetic code from it, put it into a format that can be given the same format that you get when you go to either a drugstore or to your doctor's office to get a vaccine, which is consistent with a shot in the arm that's got to be low cost of goods, no cold chain, and distributable to leverage our healthcare infrastructure. And then we would produce the antibody, the correct antibody, in that individual directly, bypassing the entire vaccine process. Vaccine process show the antigen, series of cellular signaling. Some people develop the correct antibody. We bypass it in our case. We take the recovered person's correct antibody, genetically encoded on a transient format, and then that antibody will prevent that person from getting Ebola. And I don't believe that we need the same drug concentrations that you need for therapeutics, because when disease transmission occurs, you're not getting the very, very, very high viral load that you have with diseases like HIV, influenza, Ebola, late in illness. You're getting a small inoculum at the initial. So our system will look different. Our system will not have a peak and trough. Our system will be one injection. We will maintain a drug dose that is preventive, and that drug dose will be present for long enough to be useful. We would like months to year, but it will not be permanent. Now, this is not a talk where we are providing the data results. We fund a dozen companies from the largest pharma to individual academic investigators, and we are mid-program, but I will share a couple of things. We have data with replicating RNA and DNA that we can have 4 months of expression in large animal models. We also have data with RNA and DNA that we can achieve therapeutic, not preventive, therapeutic drug doses of other monoclonal antibodies that are on the market. So in other words, we have already demonstrated 2 years into this program and 4 years into our vaccine program that we can achieve the drug levels and the duration that you would need for a product. That has not been in people yet, but we still have to find the right antibody, even if we have a delivery system. And as we showed before, there is many diseases that we have where the antibodies come up and down in abundance, just like Ebola. But even in that, there is a background of antibodies that all of us have to things we ate today, things that we are, molds in our environment, our microbiome. There's a myriad number of antibodies. So we need to be able to isolate the right antibody that's protective for that given infectious disease as quickly as possible. Now, there are 2 technologies that allow us to do this. And again, for some people in this audience, this is just common knowledge. But those 2 technologies are coming together in a way that has not occurred before. One is the sequencing of genetic information is cheap, ubiquitous, accessible, and fast. But the other is the isolation and compartmentalization of an individual biological molecule of interest that separates it from its partners. So instead of having a soup of antibodies, we are able to put into microfluidic formats 1 antibody per compartment, and we're able to sequence that antibody, interrogate it, and we're able to do that extremely rapidly and be able to demonstrate, even with the US-based Ebola survivors, that we can do this in several weeks. So when this program was ongoing, some of our investigators, because they're like all of the ones you hear today and like the people in the room, the eminent people in this field, they were contacted and wanted to participate in the DARPA program. And our investigators that were part of this program happened to be involved in this process. And the patients themselves at Emory asked to be a participation in the study as a live fire test of the system. So Almost all US-based survivors are part of this program, and all 4 of the patients that are recovered from Emory are indeed part of this program, and you're actually looking at their data. This is a facts analysis, but to keep this on a level field for all of us here technically, upper right corner are circles showing all dark dots on the 4 squares at the right side of the screen. Those are all antibodies that are binding to Ebola. The healthy person in the upper right corner, there are no presence of cells that are binding to Ebola in that individual. What we have done today, which has not been shared with the world, is taken those antibodies out. We've not only determined that they've bound, we've put them into in vitro neutralization assays, which is a nice way of saying that they prevent the infectivity of the Ebola virion. And now we have put it into animal models and showed higher potency in those models than the MAP antibodies that you may have heard about. And the MAP antibodies were derived by challenging a mouse with an antigen, giving a mouse an Ebola vaccine. And so those were mouse antibodies. And those MAP antibodies have murine or mouse sequence in them. These are human antibodies from human. And we're not going to put the antibodies in. We're going to put the genes that make the correct antibody. And that is our next step. So this is not an Ebola program. This is a program of a platform technology. Our goal here is to create a technology that is agnostic to the threat that can be deployed quickly. And it needs to scale. So with something like influenza, the scale is unbelievable because perhaps the whole country is infected. The case rate in H1N1, which is a pandemic outbreak in 2009, was as high as 6 million people per week. So the amount of transmission, essentially, you need to protect the whole country. Flu, we know how to make vaccines for. There is an enormous global market, billion-dollar market. Our best pharmaceutical companies have infrastructure in place to make flu vaccine. And using 2009 H1N1 as an outbreak with government institutions set up to handle flu, the US position to solve the problem, the fastest we moved was shipping in October 2009 after the index case in March 2009. And if you look at the area under the curve, the time that we shipped the vaccine was after the cases have already been transmitted to the greatest number of people in the population. It takes 2 weeks after vaccine, 2 weeks to develop protection. So 2 weeks after shipment, assuming that we go from shipment into arm, you have 2 weeks to be able to prevent your transmission. And then it's only 40 to 60% effective every year with seasonal flu. So we need to bypass that process. And again, I think we can do that with this type of system. So we've been working on this for flu. We've been working on this for many other common infectious diseases that are bacterial and viral and even toxins. So not only infections. Again, if we are able to gene encode and deliver without cold chain, without refrigeration, we may not be able to get that first case. We may not be able to get dozens of cases later. But I think in that early lag, we should be able to respond. And all of our proof of concepts in vitro and derived from people are showing that we should be able to do this in about 90 days. And again, we can leverage the healthcare system for vaccine because it's the same format. This is conceptually what we are trying to do. We know that there are many, many, many solutions of genetic engineering out there that are very important for mankind. But from my perspective, we are trying to look at the temporal problem of how to respond rapidly with some solutions and others of how we can respond novelly, for example, with insects, but in ways that we can do this in a safe and controlled way. And I welcome to hear all of your ideas.